Yeah, I think this is a very important study. It was just published in Blood. We have a very young, dedicated physician scientist who's been working on this project. And the long and short of it is for 5 years, we serially followed patients who were being treated on a frontline ibrutinib monotherapy trial. What we learned actually is very interesting is that, tumor cells actually use a different mechanism to overcome the BTK blockade induced by ibrutinib. What we see actually is that Lin, which is a member of the SRC family, begins to kick in and take over, and you start seeing overexpression of Lin. And we actually modeled this in our laboratory, showing, in fact, that in these ibrutinib resistant cells, by knocking down Lin, one could reintroduce, sensitivity. We've actually furthered this work now. We presented at ASH actually some very exciting models around BTK mutated disease. And what we're finding is that another SRC family member, HCK, is also cassetting in and doing actually the transmitting of survival signaling in response to MYD88 when in fact you've disabled BTK. Now, what's important to recognize is, yes, our BTK inhibitors disable kinase activity, but still there's a scaffold there that continues to function and bring in these SRC family members. And the work that we're doing right now is to try to degrade HCK in order to and BTK in order to take away that scaffold. So that work is very exciting because it's showing us that we can overcome these BTK mutations. And this is now, important across all the BTK inhibitors, not just, the covalent ones, non-covalent ones like pirtobrutinib, but also we're seeing this with BTK degraders. So we do have to target now this, upstream complex that's allowing for growth survival signaling to continue.